Hello, biology students. We are going to do a lab today called What are Proteins and How Do They Fold into Biologically Important Shapes? Remember, we looked at proteins earlier, and we know that proteins are a series of amino acids, kind of like beads on a chain. Every model has its weaknesses. This is not a real protein. These are pieces of plastic that pop together, but they can be used to represent a protein and the amino acids that make up a protein. When amino acids bond together, it does kind of function like beads on a chain. There's a backbone, and that's kind of like the pop part of the pop beads, that little spot that pops into the next one is a little bit like how amino acids bond. There's a backbone that keeps them together. And then there's 20 different kinds of amino acids. So I have 20 different different kinds of beads here, and they all bond in different ways. And some of them will start to, the side chains on the amino acids, there's a little side chain there that represents a large amino acid with some charge, can start bonding across the protein here and folding important shapes. Also, a protein's hydrophobic and hydrophilic areas can cause important shapes, and we'll be talking about that as well. These blue areas in my model here will be polar, hydrophilic, water-loving parts of the protein, and the yellow and black parts will be nonpolar parts that are hydrophobic, we call them. So we'll be using these models as well as some simulations in this wonderful Concord Consortium activity in their interactions curriculum to be understanding proteins today. So the building blocks of proteins are these amino acids. In some of my models here, they're pop beads that pop into each other. They stick to each other because they've got these little connectors. And so you can think of those connectors like the backbone of the protein. Let's explore proteins here. It says click on each C number below in order to take a closer look at how amino acids link together forming a chain we call the protein. This protein in this particular simulation is porin, which lets molecules pass in and out of cells. So we can press 1, and it'll rotate and show us a part of the protein here. And we're only going to study this very small part of the amino acid chain of this particular large protein. And that tiny chunk is 5 amino acids. Let's go to 3 here. And see how the amino acids are turning different colors. That represents 5 different amino acids. And they're linked through the backbone here, which is now colored turquoise. You can see right here, that's kind of like the pop beads popping into each other and creating a way for those amino acids to stick together. The part that branches out, these parts like the pink, red, blue, green, and black here, those are called side chains. And each amino side chain is made up of specific elements that give each amino acid different properties. And we'll be finding out more about those different properties as we move through the simulation here. Our first question here is, what is the purpose of the backbone part of an amino acid, which is the same for every amino acid? Well, we've talked about that. It keeps the protein together. Those amino acids have to stick together. And so just like my pop beads, the backbone makes it able for the amino acids to connect. So you can write a good answer to question 1. Let's stop a moment and let's talk about the bigger picture here. What are the building blocks of proteins? Well, we talked about that they're amino acids. They're important molecules that perform many functions in your body and are a part of all living things. The collagen in your ears is protein, the wiggly part of your nose, your keratin makes up your fingernails and your hair. Each protein is actually made of by linking those smaller molecules together called amino acids. And in our beads in the chain model here, these beads represent each different kind of amino acid. The protein below here sits in cell membranes. Now it's our job to find a scene that shows the side chain part of amino acid and take a snapshot. And then circle the part that is the side chain. Well, fortunately, this shows in turquoise the side chain, so we can just take a snapshot here. And I'll let you circle the side chain. And let's roll on to part 2 here. Why are some amino acids more polar than others? Because proteins are long, flexible molecules, you can see that here. In this model, you can see it in this model, and we'll get to another model here in a minute. They can not only interact with water they're typically surrounded by, but also one part of a protein can interact with another part. There are 20 different amino acids that can be linked together to make a protein. Each amino acid can be polar, nonpolar, or have a full electric charge, positive or negative. Below are 3 examples of the 20 possible amino acids. Explore their structure to understand which parts are the same and which are different. Depending on polar and nonpolar properties of the amino acids, a protein will fold into different structures. So let's read this. Explore the 3 different amino acids. Explore why different amino acids have different properties related to the charge or lack thereof on the surface of their side chains. So I'm seeing a lot of different things that we can adjust here. We can do side chain only. We can show surface charge. And we can grab these and rotate these and look at those charges. Serine looks like it has a lot of negative, that's the blue, and some positive. Valine doesn't look like it has any charge, and histidine is very negative. So if we take the surface charge off, we can look at some of the reasons for these surface charge. Serine has a big, has an oxygen here, which we know is very electronegative and kind of likes to hog or hold the electrons on its side of the bond. So it becomes negative. So that can affect the polarity of the protein. Valine has all carbon as a side chain, and carbon's very good at sharing equally, so we see that valine doesn't have a charge. Histidine here, its side chain has 2 nitrogens, and nitrogen shares okay, but it's not a perfect share, and we can see that nitrogen also creates a charge in the side chain of histidine. So you should have the information you need to explain why serine is polar. Hint, hint, there is an oxygen atom here. And why valine is nonpolar. So finish number 3, and then we'll move on to to page 3. Which parts of a protein are polar and nonpolar? Below is a protein that has an option to show each amino acid as a single bead. This view can help show how a protein molecule folds up into a 3-dimensional shape. Use this model of the protein found in egg whites to explore some of the 20 different amino acids that could be linked together to make proteins. So we're going to build proteins from amino acid components, view the components of a protein, elements in amino acids, in different colors, and zoom in to several amino acids to understand how they link together to make a larger structure. Use the mouse to drag the protein around, viewing it from different angles, and try building the proteins to see how the large molecule folds up from a particular shape. So let's look at some amino acids within this protein. Remember, amino acids are like the beads that build the long chain, and then the chain folds up based on some things we're going to talk about today. Let's look at serine. Serine has an oxygen, and we know that's polar from the last one. Phenylalanine looks pretty nonpolar. It's all carbon, it's sharing. Valine, all carbon, sharing equally. Asparagine, It's got an oxygen and a nitrogen in there. I bet that's polar. Glutamine, nitrogen and an oxygen again, probably polar. Let's show the entire protein. Those beads are kind of like the beads on the string model we're using here, where each bead represents a different amino acid. We can build this protein. You can see how the amino acids are put together here. And we can color it to see the different well, let's build it again, and we can see the different amino acids that are coming together. Those 20 different amino acids, there's 20 different colors here that are being put together to create the primary structure of the protein. Pretty cool, huh? Let's go back to the element. We can look at a couple other models here. That's ball and stick, that shows every single atom using like a ball and a stick. A stick shows the bonds and a ball shows the elements. And we can show the atoms with their electron clouds, and that's called the space filling model. We can go back to our simplified beads, and we can, I don't think we can unclick that. All right. And we can zoom in on some of these proteins. Now let's do what we need to do here. It says take a snapshot that highlights a polar part of the protein. Label a snapshot to show why it's polar. Well, this would be a good place to take a snapshot because we know there's an oxygen here, which is electronegative. And we know that the nitrogen also affects electronegativity and creates polar side chains from our last slide, or from page 1. And I think you can do question number 5 here, where you also highlight a non-polar side chain. Remember, carbon shares very well, so it's a side chain that's all carbon and hydrogen. Would probably be a good choice for a non-polar part of the protein. So pause the video, finish question 4 and 5, and we'll move on to page 4. So how do proteins fold into different shapes? We're going to use the simulation below to explore how several different proteins fold up to make a stable structure in water. As you try different proteins, look for patterns in the structures they form and where the polar and non-polar amino acids end up as they interact with themselves and with the water molecules that are not shown, but that are surrounding the protein. Now remember, water molecules like to stick together. They're polar, and when you separate them, there's a little bit of charge that develops, or a little bit of potential energy that develops from separating those charges, because those charges have pull and they like to stick together. So to get to the lowest potential energy, to get to follow the laws of the universe, which say lowest energy and maximum randomness, this protein is going to have a certain behavior that we're looking for. So we're looking for some patterns here. All polar, generate all polar, all non-polar or random protein and observe how they fold. Explore how the potential energy of the system changes over time to draw conclusions about how proteins develop stable structures. We actually can see potential energy in the next simulation on the next page. Here we're just going to look at lots of different proteins and how they fold, and notice how the polar part of the protein and the non-polar part of the protein are behaving. So let's generate a new one. Let's fold that. And be looking for patterns here about what is interacting with the blue water here and what's in the middle, not so much interacting with water. Let's generate another random one. That's a lot of non-polar. Look what happens to the non-polar as opposed to the polar that's interacting with this blue water. We don't see the molecules, but we just understand they're there. Let's generate a mostly non-polar protein. Let's see what happens with that. Interesting. Let's generate a mostly polar protein. That's doing something very different than the other models we've seen. So what patterns are we seeing? That's the question in number 6. See if you can find some patterns where nonpolar and polar are behaving. Are they behaving differently? Are they behaving the same? What happens when you fold proteins that are primarily polar or nonpolar? What do we remember about polar and nonpolar interactions from an early part of the lab to explain why these patterns of protein structure form in water? We talked about salad dressing in the first part of this lab, and we talked about how the oil in salad dressing, this is olive oil, and this is vinegar, don't always mix. We can shake it up, but when we let it sit in the fridge or let it just sit out on the countertop, the oil and the water don't mix. They'll separate back out. Just like the hexane in water didn't mix in part of the lab. The hexane, remember, is clear, and the blue, I dyed the water blue. If we shake this up and we let it sit, it will separate out again because the nonpolar hexane doesn't mix with the polar water. So we can use what we know about polar and nonpolar interactions from the first part of the lab to explain what's going on here with our simulation. So I'll let you finish questions 6 and 7. You can pause the video, and I'm going to go on to page 5. So now that you've answered questions 6 and 7, let's talk about what do protein folding patterns depend on. Amino acids can be grouped into 2 broad categories: polar, or charged, and nonpolar. The polar and charged ones are called hydrophilic, which means water-loving. And in this particular model of proteins, we're going to have the blue be hydrophilic, or water-loving. It's easy to remember because blue, water, they get along. Water is blue. So this is the part that's going to want to interact with water because it's polar. The nonpolar ones are going to be called hydrophobic. And in this model, they're black and yellow. That means water-fearing. And I made these black and yellow to think of, there's a lot of scary things in nature that are black and yellow. So there's a phobic thing, and this is hydrophobic, which means basically scared of water. In this simulation, you can create your own protein by clicking on an amino acid to change it to one you selected from the list of possible amino acids. Try creating several different proteins and observe where the different types of amino acids end up and what protein folds in a water environment. So here's our simulation. How do peptides form stable conformations? Generate all the hydrophilic and all the hydrophobic or random proteins and observe how the protein folds in response to these intermolecular properties. Explore how the potential energy of the system changes over time to draw conclusions about how proteins develop stable structures. So let's just start out. We've got a new random protein, and let's click start and see what happens. Look at what's going on with the potential energy, and notice patterns with the hydrophilic or water-loving polar proteins here, and the hydrophobic yellow proteins or non-polar amino acids that are in the middle. Let's generate a new protein and see if that pattern continues. Look at what's happening to the potential energy here. We've got the polar or hydrophilic water-loving proteins, or amino acids on the outside. And we have the hydrophobic amino acids, which are non-polar, right here. Let's keep seeing if we see the same pattern over and over. There's some interesting things going on here. Pay attention to potential energy. I'm going to reset it. Look at the potential energy. I'll start it and watch what happens to the potential energy over time. I'm going to reset. Watch what happens to the potential energy right here over time. So how does the strength of polar and nonpolar amino acids interacting with each other and water molecules drive protein folding? You can think of it as, remember we talked about water molecules, if they're separated, anything charged, anything polar, that separation creates some potential energy, and when they go back together, there's no more potential because that force that's pulling them together has already pulled them together. So that same thing, if the water-loving or polar parts of the protein want to be with water, they're going to be with water together. And you can see what's happening to the inside, to this hydrophobic parts of the protein. I have to kind of push them together so I can get water, the water-loving, the hydrophilic parts of the protein on the outside of my model here. I want water on the water-loving, the polar proteins on the polar amino acids on the outside, and I want to put those non-polar away from the water so it has the hydrophilic amino acids, those hydrophilic parts, water-loving parts of the protein, can have the room on the outside to interact with water, because that's going to lead to the lowest potential energy. So we have a couple snapshots to take here. Take a snapshot and add labels to show your understanding of how amino acids interact with each other and with the surrounding water molecules to cause the protein to form this shape. We need to take another example of a protein, and just so we have more examples here, we have an all hydrophobic protein. Look, notice potential energy here. I'm going to reset that. Oops. Generate an all nonpolar protein. Start that. Watch what happens to the potential energy. And then let's compare it to an all polar protein. We've got this potential energy. Watch what happens. And watch what happens to the shape. They're very different when it's nonpolar versus all polar. And also keep your eye on that potential energy. If you want to, we'll be able to generate our own protein in the next page. So the term hydrophobic means water-fearing. This definition works because it describes the overall behavior, but there's something misleading about it. Why doesn't water-fearing describe what's happening? If you think about the forces between polar water and non-polar molecules, if I'm trying to lower the potential energy of my particular model here, and in order to do that, I want my hydrophilic, my polar parts of the protein to be out here interacting with water on the outside. In order to do that, I kind of have to shove everything else to the middle. And it's not that the hydrophobic or the non-polar parts of the protein, those non-polar amino acids, it's not that they don't like water, it's just that they get pushed around. In order to minimize the total energy here, to minimize the potential energy, we have to do some things to that those non-polar amino acids, those hydrophobic, in order to make room for the hydrophilic, the water-loving, to be on the outside interacting with water, because water and the polar amino acids, the hydrophilic amino acids, want to interact with each other because that lowers the potential energy of the system. And having a lower potential energy of the system is just a rule of the universe. Minimum energy and maximum randomness are laws of thermodynamics that govern chemistry and physics and a lot of biology as well. I think we just went over to some of the answers. Remember, potential energy is higher when polar molecules are separated, And when they get together, the potential energy goes down. We just talked about the law of nature that says things have to trend towards lower potential energy because it's a rule of the universe. And how does that lead to stable structures? If we want to lower the potential energy of our protein model here, we let the polar, the hydrophilic, the water-loving amino acids be on the outside in order to get them together and lower that potential energy. All right, finish through question 14, pause the video, and we'll work on the next page here. What do proteins look like? The previous two simulations showed a simplified view of proteins, only two-dimensional, to help us illustrate certain folding patterns. However, the interactive below shows a more realistic three-dimensional protein structure. Explore these to see if you find similar patterns how real proteins fold into stable structures. Let's look at this. Exploring polar and nonpolar regions of different proteins. Select different proteins and choose which part of the protein molecule to highlight. Click and drag the molecule to rotate. Observe where the polar and nonpolar parts of the protein end up and how they influence the overall structure of the larger protein molecule. All right, let's check it out. I always just like to go in and play around with a protein before with a model before I try to do anything with it. So we've got different proteins here. We can make the nonpolar parts of the protein translucent, so see-through basically, so we can just see the polar parts. We can make the polar parts invisible or almost invisible, so we can just see the nonpolar parts. That's important in recognizing what's on the outside and what's on the inside and some of the other structural features. I'm going to put it back to none here. We can put it on ball and stick model. We can rotate it around here and see what's going on in the back and the sides of the protein. This is the space filling model where the electron clouds are shown. We can just go by the colors of the amino acids. That just is a little bit mind-blowing because you can see the 20 different amino acids in there. We can go on elements. We can see how many oxygens are hanging out on the outside and how many nitrogens on the amino acid side chains are showing. And we can rotate that around. Or we can look at polarity. Remember, green is polar and yellow is nonpolar. So if we click through these proteins, are you noticing a pattern of where polar is versus nonpolar? Here's question 15. Take a snapshot that shows a pattern found in all of the proteins, showing how the protein folds and where the polar and nonpolar parts of the protein end up. Explain why this pattern occurs by talking about forces and energy. So I'll let you take a screenshot and you can choose which protein you want to take a picture of. Make sure you do the reading here. And we're done. Thank you so much to Concord Consortium for this excellent lab that's part of the Interactions curriculum.